Hi everyone, welcome back. In this video, I'm going to talk about lipoproteins. So basically, what are chylomicrons, VLDL, LDL, and HDL, and then we'll go through how each of them are going to transport lipids in the body between our different organs. So let's get started. All right, so let's start with a quick background about lipids. So in our body, we have three main types of macronutrients, and these are things that our cells can break down to generate ATP or to use to synthesize things. And these three things are the proteins, carbohydrates, and lipids. And some of the common examples of lipids that are going to circulate in our body are triglycerides. So these are the storage forms of fatty acids. Cholesterol, which gives our membrane fluidity and is also used to make steroid hormones. And then phospholipids, which are the main things in our cell membrane. And so in this video, we're going to focus on the lipids right here and how they're going to be transported around in the body to get to the cells where they're going to need to be used. And the key thing here to keep in mind is that while these proteins and carbohydrates are hydrophilic, that means they have no problem circulating in blood. Lipids are hydrophobic. So this is going to present a special challenge because blood is mostly water and these guys don't like water. So we need to find a way to transport them around in the blood. And this is where the lipoproteins are going to come in. All right, so and to solve this problem and make our lipids able to circulate around in the blood, we have to attach proteins on their surface. And so just imagine that we have this kind of big ball of lipid right here. So it has, triglycerides, cholesterol, phospholipids, and it can't swim. It's scared of water or hydrophobic. So what we can do is just give it some floaties so that it can swim in the water. And these floaties that are the blue things that are on the surface are the proteins. So these proteins are going to attach to the surface of these lipids and allow this to be able to circulate in the blood. And the name of these proteins, these are called apoproteins and also known as apolipoproteins. So sometimes people get confused with the naming. So if it starts with an apo, it's just referring to the protein that is on the surface of these lipoproteins. And so what's going to happen together now is when you add these lipids to these proteins, these are called lipoproteins. So you have the lipid in the center and the protein, the apoprotein or apolipoprotein, same thing on the surface. And finally, these proteins aren't just floaties. They also have two more functions. Number one, these apolipoproteins are going to bind receptors. And number two, they're also going to activate enzymes. And so they're going to be super important in the processes. And we'll see that in more detail as we check out each of the processes going forward. But this is an overview of what lipoproteins are and what they're made up of. All right, so there are actually different types of lipoproteins, and there are actually 5 different ones. So we have chylomicrons, very low density lipoproteins, intermediate density lipoproteins, low density lipoproteins, and then finally high density lipoproteins. And as you're going from left to right here, so from chylomicrons into this direction towards HDL, they become smaller in size and more dense. So that means that chylomicrons and VLDL are the biggest ones and they have the lowest density, and HDL, for example, is the smallest in size and it has the highest density, and that's why it's called high density lipoprotein. But the other thing that's important to know is that also as you go from left to right, the amount of triglyceride inside each of these becomes less. So again, for example, chylomicrons and VLDLs, these are mostly triglycerides. You can see all the yellow inside of them. And then LDL, for example, you can see that that's mostly cholesterol, and that's all the red right here. So red corresponds to cholesterol, yellow corresponds to triglycerides. And then HDL is mostly proteins. You can see all of the green right here. And so each of these lipoproteins is different in their composition, and that's because they each have different functions. So now we're going to go through each of them one by one and learn about each of them in more detail. All right, so let's start with chylomicrons. So these lipoproteins are made by the enterocytes. And remember, enterocytes are the cells that line the small intestines. And so the source of the lipids here are going to be food in the diet. So basically what's going to happen is that, let's say you eat a slice of pizza, it's got lots of lipids. These lipids are going to get broken down, digested in the GI tract. And then these lipids are going to get absorbed into those enterocytes. And then inside of them, they're going to get packaged into these chylomicrons right here. And remember that we said earlier that the main lipid inside of the chylomicrons is going to be triglycerides. So basically you can think of these chylomicrons as little balls of mostly triglycerides, which have been absorbed directly from our diet, straight from our food by the small intestines. And so then the role of chylomicrons, the reason for having them is that they can get secreted into the circulation and then deliver these dietary triglycerides that we just ate and absorbed into our peripheral tissues so that our cells can use them. And then the final point about chylomicrons is that they have 3 apoproteins on their surface. We have ApoE, ApoC2, and ApoB48. And from these 3, the main unique one is ApoB48. That's only on chylomicrons. All right, so now let's move on to very low density lipoproteins, or VLDL. So these ones are made and secreted by hepatocytes, which are liver cells. So remember that one of the many jobs of the liver is to make and store lipids. And so it can deliver it to the other cells. Remember, it's called the liver because it delivers. So the lipid source right here is going to be lipids that were synthesized by the liver. And just like chylomicrons, VLDL also has both triglycerides and cholesterol, but more triglycerides, just like we saw with chylomicrons. So the main role here is going to be to transport these triglycerides that were made in the liver to peripheral tissues. And this is really important, right? Because we're not constantly eating food every minute of the day. I wish we were, but we're not. So for all those times in between, when we don't have circulating chylomicrons, we're going to need another way to circulate fat around the body to the cells that need it right away, and that's the role of VLDL. So it's going to circulate these triglycerides to our cells so that they can use them. And so the final point is that VLDL also has 3 apoproteins on its surface. So it has C2 and E, just like we saw with chylomicrons, but instead of B48, it has the longer version, which is a different apoprotein called B100. All right, so next is intermediate density lipoproteins, or IDL. And this is actually where it gets really interesting because IDL isn't synthesized or secreted by any cells. It's actually formed right in the bloodstream from the degradation of VLDL. So what happens is that as triglycerides are removed from VLDL by peripheral tissue, what's going to remain of that VLDL is going to have much less triglycerides because they were removed. So the ratio of triglycerides and cholesterol is roughly the same, and this is IDL. So basically, IDL is what's left over from the degradation of VLDL in the circulation as those triglycerides were removed from it. And so one of 2 things is going to happen to this IDL. About half of it is going to get taken up by the liver, so removed from the circulation, and the other half is going to get further degraded and turn into the next lipoprotein, which is LDL, and that's exactly why this is called intermediate density lipoprotein, because it's the intermediate or the middleman between VLDL and LDL. And then finally, as far as apoproteins go, it's got the same ones as a VLDL had on it minus ApoC2, so that means the ones that still remain on IDL are ApoB100 and ApoE. All right, so next is LDL, or low density lipoprotein, and we kind of already talked about this one a little bit. LDL is made from the further degradation of IDL, specifically in the hepatic circulation. And over there's an enzyme called hepatic lipase that's going to cleave most of the remaining triglycerides off of IDL. And so as this happens, what's going to be left over is going to have very little triglyceride left in it. And so the ratio is going to be much higher cholesterol than triglycerides. And this is LDL, so it's mostly cholesterol. And so its role then is going to be to deliver this cholesterol that was synthesized in the liver, and remember, originally secreted as VLDL, to the peripheral tissues so that the cells can use it. And then finally, as far as the apoproteins go, when we get down to LDL, we've also lost our ApoE. So the only one left on LDL is ApoB100. That's the main apoprotein of LDL. All right, so the final one then is going to be HDL, or high density lipoprotein. And this one isn't part of the VLDL, IDL, LDL thing that we talked about. This one's totally separate. It's actually made by the liver and to a lesser extent by the intestines, and it's secreted by them into the circulation, and it's mostly made up of just proteins, so not a lot of lipids when it's first secreted, and that's because its main role is to actually go out there and pick up extra lipids, especially cholesterol, from the peripheral tissue. And this is actually super important because if this doesn't happen, then all of that extra cholesterol is going to accumulate in places like the lining of our blood vessels, and it will turn into plaque and cause atherosclerosis and complications like MI and stroke. And so this is why HDL is called good cholesterol, because its job is to go out there and remove or scavenge extra cholesterol and bring it back to the liver, so getting it out of the circulation. And so this is going to reduce the risk of plaque formation and atherosclerosis. Now, the other main job of HDL is to serve as what's called a repository for ApoE and ApoC2. And all this means is that HDL is the thing that donates these 2 apoproteins to chylomicrons and VLDL. And so the main apoproteins then that you're going to find on HDL are going to be ApoE and ApoC2. And that's because it can donate them to chylomicrons and VLDL. And then the other one is ApoA1, and this is the one that's unique to HDL. So HDL is the only lipoprotein that has A1, and you can remember that because A is a good grade and HDL is the good cholesterol, so HDL is the only one that gets the A grade. All right, so that was a basic introduction to our 5 lipoproteins. And so the next thing is now to learn about these processes that they're involved with and the way that they're going to transport it. Basically, you can break it down into 3 processes that are happening at the same time. So the first one is the chylomicron transport. Remember that this is being secreted from the small intestines into the circulation. The goal was to deliver triglycerides to our tissues, and then after those triglycerides have been removed, whatever is left over is called the chylomicron remnants. That's going to be taken up by the liver. So it's going to start in the small intestines, deliver triglycerides to tissues. The remnants are going to be taken up by the liver for storage. The second transport process started as VLDLs, and remember these were secreted by the liver into circulation. And again, the goal was to deliver triglycerides to tissues. And what's going to be left over is IDLs, which were the intermediate density lipoproteins. And then more triglyceride was going to be removed. And then what's going to be left over was the LDL, or low density lipoproteins. And then they were going to deliver cholesterol to the tissues and then eventually return to the liver and be removed. And then finally, the last transport is HDL, or high density lipoprotein. Remember, these are secreted by the liver and small intestines. And so they go into the circulation and they have the opposite function. So their main goal is to remove excess cholesterol and other lipids from the periphery, and then also help out the chylomicrons and VLDLs by donating them those 2 key apoproteins. And so these are the 3 key processes. And now let's look at each of them in more detail. All right, so let's start with process 1, which was the chylomicron transport. So again, chylomicron being released from the small intestines, getting into circulation, delivering triglycerides to tissues, remnants returning to the liver. So let's start over here. We have our small intestines and the lumen of the small intestine right here. And so here we've ingested our pizza, the fat's broken down and emulsified into my cells, and then the fat's going to get absorbed by the enterocytes. So these are the villi of our enterocytes through the brush border. And so the fat now is going to get into this enterocyte right here. And so what's happening in step 1 right here is that this dietary lipid that got into the enterocyte is going to get packaged into chylomicrons. Now, these chylomicrons are too big to go directly into blood, so they actually get secreted into lymph first. And then they're going to circulate around in lymph channels, and then eventually from the lymph channels, they're going to enter the large blood vessels, such as the subclavian vein. They're going to enter the systemic circulation of blood. So in step 3 right here, now they've gone from the lymphatics into blood, and now they can be delivered all around the body. And so then what's happening in step 4 right here is that in the peripheral tissue, like in adipocytes and muscle, these triglycerides are going to be removed from the chylomicrons. And so now you're going to have fatty acids delivered to the adipocytes and muscle cells. So basically our peripheral tissue. And then what's left over after these triglycerides have been removed is the chylomicron remnants, because they're the remnants of the chylomicrons. And so in step 5 right here, they're going to be taken up. And so basically what we accomplished here was that we could absorb the dietary lipids into our body tissues so that these organs can use them for use or for storage. All right, so now let's look at this same exact process of chylomicron transport, but this time we're going to add in the enzymes and the apoproteins that are important for making this happen. So we're going to add in the final details that are necessary for this process. So again, starting over here in the beginning where we have the fat absorption into the enterocytes, we have the formation of the chylomicrons, and now we have an apoprotein that's added here. This is ApoB48. So remember, ApoB48 was the one that was unique to chylomicrons, and this is really important because this ApoB48 needs to be on the chylomicrons for it to be sort of recognized as a legit chylomicron and to be secreted into the lymphatics. It will not get secreted into lymphatics if it doesn't have B48. So at this point, it only has ApoB48 on it. It gets secreted into the lymphatics, and then it goes into blood, as we talked about before. And then in blood, it's going to get ApoC2 and ApoE from HDL. So remember, a few slides back, we said one of the roles of HDL was to serve as this repository for ApoC2 and ApoE. And so now you can see that this is how chylomicrons are getting it into circulation. It's going to get donated to them from HDL. So that's the second main point right here. And then, so again, we're just going to continue what we talked about before. The chylomicrons are now in the peripheral tissue. And remember, the triglycerides are going to be taken off the chylomicrons and delivered in the periphery to the adipocytes and muscle cells. And the way that this happens is by an enzyme called lipoprotein lipase. So LPL stands for lipoprotein lipase. And this enzyme requires ApoC2 as the cofactor for it. So ApoC2 is the cofactor for lipoprotein lipase, which is the enzyme that is important for cleaving triglycerides off of chylomicrons in the periphery. And so the final point here is that as the chylomicrons are getting degraded and the triglycerides are being removed, the other things that are being removed are B48, ApoB48, and ApoC2. And so the only thing that remains on the chylomicron remnants is ApoE, and this ApoE is important because it helps facilitate the uptake of these remnants out of the circulation by the liver. All right, so now let's talk about process 2, which is the transport of VLDL. And so again, as a quick recap of our overview of this process, you have VLDL being secreted from the liver. It's going to circulate in the bloodstream, and it's going to deliver triglycerides to the tissues. So then it's going to become IDL, and then that one's going to lose more triglycerides at the liver, and it's going to turn into LDL. Then LDL is mostly made up of cholesterol, so it's going to deliver this cholesterol to the tissues. Then finally, it's going to be pulled back out of the circulation by the liver. So now let's look at the steps in this process. So we're starting in the liver because this is where VLDL is made, synthesized, and secreted, and it's going to be in step 1 here, secreted directly into blood. So this is one important difference between VLDL and chylomicrons, because remember that chylomicrons were secreted into the lymphatics first. Here with VLDL, it's being secreted directly into blood. So that's one important difference. And then this VLDL is going to circulate around in the blood until it reaches the periphery, so things like adipose. And so the main thing that's going to happen to VLDL is that the triglycerides are going to be removed, because VLDL is mostly made up of triglycerides, and so the main goal here is to give off some of these triglycerides to the periphery so that they can use it. And when this happens, when the triglycerides, a large number of them have been taken off VLDL, right here in step 2, then the product of that, or what remains, is IDL. And then this IDL is going to continue to circulate and until it hits the hepatic circulation, and there it has two choices. So half of it gets uptaken back by the liver and pulled out of the circulation, and the other half, you'll have the removal of more triglycerides off of that IDL. So more triglycerides get cleaved off into the liver, and what remains then, when at this point most of the triglycerides have been taken away, is LDL. And so then this LDL is going to continue to circulate around in the bloodstream, and it's going to go to our peripheral cells, and there it's going to deliver cholesterol to those tissues so that they can use it, because again, LDL is mostly made up of cholesterol. And then whatever remains of the LDL is going to then return back to the liver and get taken out of the circulation by the liver. And in this way, the entire process is complete. So we start by secreting the LDL, then it gets converted to IDL, which then becomes LDL, and then that LDL gets reuptaken back by the liver. And so that's all the 4 steps that's outlined here. And the final point is that the goal of this is for the liver to deliver its goods, which are the lipids, to the other cells. So it's given the triglycerides over here so that they can use it, and it's giving the cholesterol over here so that they can use it. So in this way, you can think of this VLDL pathway as the Robin Hood. It's stealing from the rich, which is the liver, and giving it to the poor, which is the other cells that need these lipids. All right, so now just like we did with process 1 with chylomicrons, we're going to do the same thing with process 2 with VLDL transport by adding the enzyme and the apoproteins to complete the whole picture, right? So let's start over here again. So starting with VLDL in the liver, you'll notice now there's an apoprotein on VLDL, and this is ApoB100. So B100 is the key apolipoprotein that's going to start off on VLDL as it's being synthesized, and now it can be secreted into blood. Then what you notice here now in this step is that in the blood, now it's picked up the 2 additional apoproteins. It's picked up ApoC2 and ApoE. And where did these come from? They came from HDL. So just like we saw with chylomicrons, HDL is going to donate ApoC2 and ApoE to VLDL as well. And that's the next important thing. And then as this VLDL now continues here, remember the next step was the cleavage of the triglycerides in the periphery. And this cleavage of the triglycerides is once again mediated by lipoprotein lipase. So this is the enzyme, and this enzyme requires the cofactor, which was ApoC2, to allow for this cleavage to happen. So again, just like we saw with chylomicrons, this cleavage of triglycerides by lipoprotein lipase and the need for ApoC2 as the cofactor for this cleavage in VLDLs is the same as we saw in chylomicrons. So these are the key points with VLDL. C2 and E are donated from HDL, and C2 is the cofactor for triglyceride cleavage by lipoprotein lipase. So now let's continue on because after this triglyceride has been taken off, then what remains is IDL. And what you also notice is that in the meanwhile, we lost ApoC2. So that was our cofactor that was, you can think of it as having been used up over here. So now what remains is ApoE and still B100 on IDL. And so now what's going to happen in IDL is, again, some of it is going to be uptaken by the liver that we talked about before. And this uptake by the liver of the IDL is mediated by ApoE. So this ApoE does actually play a role. It's important for uptake by of lots of these remnants by the liver, including of the IDL. And remember, the rest of the IDL that doesn't get taken up was going to be cleaved here. So the triglycerides were going to be cleaved off of it at the liver, and that's how IDL becomes LDL. And this cleavage of triglycerides is once again going to be mediated by a lipase, but this one is called hepatic lipase. And that's because unlike this other lipase, this hepatic lipase is only present in the liver circulation, and that's where this cleavage of IDL to LDL is happening. So the key point here for IDL is that the triglyceride cleavage is mediated by hepatic lipase, not lipoprotein lipase, and hepatic lipase does not require a cofactor. So lipoprotein lipase required C2 as a cofactor, hepatic lipase does not require a cofactor. And then the final thing that's left over now is LDL. And so LDL only has ApoB100 on it. And this is really, really important because this ApoB100 is actually what binds LDL receptors. So the way that LDL is taken up by either the peripheral cells or by taken back up by the liver at the end of its journey is by the binding of ApoB100 to LDL receptors. And when this binding happens, it's going to activate what's known as receptor-mediated endocytosis. So the LDL is going to be endocytosed into those cells and taken up. So the key point for LDL right here is that its interaction is mediated by B100 binding to the LDL receptors, causing receptor-mediated endocytosis. And this is going to happen both on the target tissues, which are these peripheral cells to use the cholesterol, or by the liver, which is going to remove the LDL from the circulation. All right, so now let's move on to our final process, which is process 3, which is HDL transport. And just like we did with the other two, I'll start with a quick overview of what's happening here. So we have HDL being secreted from the liver and some from the small intestines. It's going to circulate in the blood. It's going to do its functions, which are scavenging cholesterol from peripheral tissues, donating apoproteins, and exchanging some lipids. And then it's going to be taken back up by the liver at the end. So now let's go through the steps here. So step 1 right here is the synthesis and secretion of HDL. And as we just talked about, it's mostly done by the liver, but a little bit of it is made and released by the small intestines. So that's what we're seeing right here. And just like we saw with VLDL, HDL is also secreted directly into blood. So this one doesn't need to go through lymphatics either. It's getting secreted directly into the blood systemic circulation. What you're going to see in step 2 is one of its main functions was to donate apoproteins, specifically ApoE and ApoC2, to chylomicrons and VLDL. So here our HDL is donating these. And one thing I forgot to mention is that I called this HDL-N. That's what it's called. N stands for new or nascent, and that's because that's the initial immature HDL that has been secreted. So that's what we're dealing with right now. But what's going to happen is that in step 3, this is the other function and the main function that we think about for HDL, which is that it's scavenging or pulling off cholesterol from peripheral tissues and packing it inside its core. And when it does that, it goes from HDL-N or nascent to HDL-M, which is mature. So now it's matured because it's done its job. It's pulled off the cholesterol from the peripheral tissues and packed it inside of it like a nice little suitcase. And then the 4th step here is that one of the important functions of HDL also is that it can actually exchange lipids. So it can do a little bit of trading before it comes back. And basically what it tells chylomicrons and VLDLs is, hey, I have a lot of cholesterol inside of me now. I have a lot of these cholesterol esters. How about I give some of this to you guys, and you guys give me some triglycerides, because I know you two are packed full of triglycerides because that's what you're made up of. And in this way, HDL can also take back some triglycerides with it. And so in step 5 now, our HDL, which is our mature HDL, has scavenged all of this cholesterol from peripheral tissues, taken some triglycerides from these guys, and is returning it back into the liver to get it out of the circulation. And so the goal here is to remove these lipids from peripheral tissues. And also remember, the other function is to give these 2 important apoproteins to chylomicrons and VLDL. All right, and so finally, just like we did with the other two processes, let's do HDL transport by adding enzymes and apoproteins to it to talk about it in all of its detail. So remember, HDL has these three apoproteins on it, ApoA, C2, and E, and so here it is right here. And the reason that it has ApoC2 and ApoE on it is so that it can donate them to chylomicrons and VLDLs. And then ApoA1 is important because it activates an enzyme called LCAT. LCAT stands for lecithin cholesterol acetyltransferase, and this enzyme is really important because this actually performs this function of cholesterol esterification in the peripheral tissue. What cholesterol esterification does is you can kind of think of it as packing your clothes inside of the suitcase, so it allows the cholesterol in the peripheral tissue to be easily packed inside of the HDL so it can be scavenged. So this is the critical enzyme. LCAT is the critical enzyme that allows for the scavenging or packaging of this cholesterol inside of the HDL, and it requires A1 as its cofactor. So if you have a problem with LCAT, if you have a mutation in LCAT, or if you have a mutation and you don't have A1, then this is going to basically make the HDL particles non-functional and unable to scavenge cholesterol and do reverse transport and bring it back to the liver. So this is a very important enzyme for the function of HDL, and it requires A1 as its cofactor. That's why HDL has A1 on its surface. And then the other thing that's important is that, remember, one of the other functions of HDL was to exchange lipids with VLDL and chylomicrons by exchanging cholesterol esters with triglycerides. And the enzyme that mediates this, you need to know, and this enzyme is called CETP, and CETP stands for cholesterol ester transfer protein, and you can think of this as, that's its function. It's transferring cholesterol ester to VLDL and chylomicrons, in exchange taking away triglycerides. So that's the second important enzyme associated with HDL. So LCAT's important for cholesterol esterification and scavenging cholesterol. CETP is important for the other function, which is the exchange of lipids between HDL and these other lipoproteins. And then finally, the last thing that's important is that ApoE on HDL is required for the reuptake of the mature HDL by the liver back from the circulation. So just like most other lipoproteins, HDL also needs the ApoE on its surface so that it can be removed from the circulation by the liver at the end of its journey. All right, so now you learned all about the different lipoproteins, what they're made up of, and how they're transported. So now it's a good time to see how this applies clinically. And one of the main easiest ways to think about this or apply this is when you're ordering lipid panels on the patient, you're going to commonly hear the terms bad cholesterol when people are talking about LDL versus good cholesterol when people are talking about HDL. And now you can kind of understand why this terminology is used, right? Because LDL has the highest cholesterol concentration among all the lipoproteins. And remember, the job of the LDL was to take that cholesterol from the liver and deliver it to peripheral tissues. So what that means is that if someone has really high LDL levels, that means they're going to get a lot more cholesterol delivered and potentially deposited in their tissues, such as the walls of the arteries right here. And so what that can do is all of this can lead to atherosclerosis, as shown in this picture, which can lead to formation of plaque, which can then cause things like cardiovascular disease like MI and stroke. And in fact, this is the #1 cause of death in the US. So that's why LDL is known as the bad cholesterol. So if its values are high, that's concerning and increases the risk for all of this. So on the other hand, HDL is good cholesterol because, remember, it does the opposite. The job of HDL is to actually scavenge and pull away cholesterol from peripheral tissues, such as from our arteries and other places, and reverse transport that. That means bring it back from periphery to the liver. And in this way, it's going to reduce the amount of cholesterol there and therefore reduce the risk of plaque formation and complications. And how this might tie in is that it's been shown that possibly light ethanol consumption may actually increase the levels of HDL by promoting its synthesis in the liver, and therefore having, for example, a glass of wine for dinner might actually reduce the risk of cardiovascular disease by increasing the levels of HDL and promoting this reverse transport. All right, so that's all the concept that we're going to learn in this video. What I'm going to do in the next couple of minutes is just recap the things that we learned by going through these high-yield summary tables for lipoprotein transport, specifically focusing on the different enzymes and then the apoproteins. So there are 4 important enzymes that are involved in this process. The top 2 are the lipases. So first one was hepatic lipase. the function of this one was triglyceride hydrolysis in the liver, and the way you know that is because hepatic means liver, so this one is located in the liver, and specifically the only place that this was involved was removal of triglycerides from IDL, which then turned it into LDL. The other lipase is lipoprotein lipase, and just like all lipases, the job of this one is also triglyceride hydrolysis, except this one, lipoprotein lipase, was in the periphery, not in the liver, because it's not the hepatic lipase. And this one then was important for triglyceride removal from two other lipoproteins, which were chylomicrons and VLDL. And the thing that's important to remember about this one also is that this one requires ApoC2 as a cofactor. So the key differences between these two is their location, periphery versus liver, the apoproteins they're lipoproteins that they act on, IDL versus chylomicrons and VLDL, and the fact that lipoprotein lipase requires ApoC2 as a cofactor, hepatic lipase does not require a cofactor. The other two enzymes are not lipases, so their job isn't triglyceride hydrolysis. So first one here is LCAT. This one was important for esterification of cholesterol in the periphery for HDL, and so it's very important for packaging cholesterol inside HDL and allowing reverse transport to happen. And the other thing important to remember here is that LCAT requires activation by ApoA1, which is on the surface of HDL. And then the final enzyme here is also important for HDL. So both of these bottom two are specifically related to HDL only. And that last enzyme is CETP, cholesterol ester transfer protein. And this one's important for exchanging lipids between HDL. So HDL trading cholesterol for triglycerides with the other lipoproteins, such as chylomicrons and VLDL. All right, and so we talked about the enzymes. Now let's wrap it up by talking about the apoproteins. There are 5 main apoproteins, so I'll go through them in order. ApoA1 is only on HDL. Remember, HDL is the good lipoprotein. A is a good grade, so A1 is on HDL. The main role of A1 is to activate LCAT, which is a crucial function of HDL to allow scavenging of cholesterol. Then we have B48. B48 is only on chylomicrons, and it has a very important role. It needs to be on those chylomicrons before they can be released from the small intestinal cells into the lymphatics. The next one is B100. B100 is on VLDL and both of its degradation products, which are IDL and LDL. And the key role for B100 is that it's actually the ligand on LDL that binds the LDL receptor and mediates that receptor-mediated endocytosis. So this is the key function of B100 to allow uptake of LDL by peripheral cells as well as backup by the liver. Next one is ApoC2. That one's on chylomicrons, VLDL, and HDL. The reason we have it on chylomicrons and VLDL is because it's the cofactor for the cleavage of triglycerides by lipoprotein lipase. So it needs to be on these two to be able to activate lipoprotein lipase. The reason it's on HDL is because HDL is actually the thing that donates ApoC2 to chylomicrons and VLDLs. So that's why ApoC2 is present on these 3. And then the last one is ApoE. Remember, E for everything except for LDL. So ApoE is important because it's on all the other lipoproteins, and the main function of it is that it allows uptake of their remnants by the liver. So it allows the liver to clear the remnants of all the other lipoproteins except for LDL, because LDL uses B100 binding to the LDL receptor to be taken up. Everything else uses ApoE. All right, and so here's a final summary slide capturing everything we talked about. These are the 5 lipoproteins, the source where they're made or secreted, and then finally the action of each of them. I'm not going to go through everything here, but you can feel free to use a slide to help you study. Okay, that's it and that's all. If you want to smash that like button or just hit it gently. If you like this video and you enjoyed it, please like it, comment, or share it with others. Subscribe to the channel so that you can keep up with all the content. And I'll link some additional videos that are related to this, specifically about the familial dyslipidemias, so that you can watch those next. But for now, thanks for watching, and I'll see you next time.